One thing I would like to let you know is that even though myself and Jerome and possibly Nick didn't give our lecture yet, you will have all our presentation. I see people taking pictures, so you will have all of that. And, okay. So we will make sure we give it to you. So, as you know, the incidence of seizure in neonates is very high, and is higher than in any other period of time. And of course, this is of concern in the NICU, and this is a neonatal and medical emergency. And this is because the neonatal brain responds with seizures to many different insults, especially acute insults, such as hypoxia, ischemia, infection, metabolic disorder. This is really the first symptom, the first neurological symptom of several insults to the brain. So what do they do to the brain? There are many papers, and this is one of the first of Mark Scher in 1993, showing that the earlier the age of onset of seizures, the higher the likelihood of neurological sequelae. Means that if you have seizure when you are a newborn, there is a high possibility that you can have additional damage. And particularly, about 40% of their infants who have seizure, they will be subsequently handicapped, and this is almost increased to 90% in preterm babies. What is also shown is that, and this is also a nice paper, an old paper, that shows that the full scale of IQ decrease with the time of seizures during the first year of life. And this is a paper that has been published in 2011 of epilepsy, very nicely showing that if you take 100 patients with intractable epilepsy secondary to focal brain lesion, so when you have already a structural lesion, but it showed that the age at seizure onset was associated with low IQ score, and particularly if you start to have your epilepsy at before 24 months, before 2 years of age, your full-scale IQ will be 14 points below what is seen in patients that starts after 2 years of age. And this is a very nice work recently published on neurology by the group of UCSF. Stephen Miller at that time was there, showing that in babies with perinatal asphyxia who present with seizures, and in this work they consider only clinical seizures. I don't know if we have a pointer. Anyway, with with clinical seizures, the severity of damage was increased in patients with seizure. It means that they studied this with magnetic resonance spectroscopy, as Jerome was showing you before, and they showed that if you have perinatal asphyxia and you have structural damage, but if on top of it you have seizures, the seizure-associated brain injury shown with MRS is larger than the structural damage. And this is what they've shown, so this is their work, so no seizures, and you have an higher NAA level and lower lactate, and if you have seizure with the same injury impact, structural injury, your lactate peak will be higher, and so your damage, particularly in the basal nuclei and intervascular boundary zone, will be more extensive. But if you look at clinical work that took in consideration all seizure in neonates, and this is a nice paper by Ronan, he showed that about 50% of newborn with seizures, they actually do well. And if you consider that there is this disorder, which is a rare genetic epilepsy on the newborn, in which neonates, they start to present with seizure on day 2 or 3. Seizures are brief, but they can often go up in frequency, and the baby can go into status epilepticus. They can seize for 2 days, 3 days, 4 days. Mean cluster is up to 3 days, but they actually do very, very well. They have normal neurological outcome. They have no epilepsy or only few patients have epilepsy later in life. So what do we know? We know that seizures during the neonatal period are associated with adverse neurological outcome, but also we know that some babies, they can have a lot of seizure and have excellent outcome. So it is still unclear, and there is a lot of work going on in the past 20 years to determine whether seizure per se can contribute to the outcome. So we have questions from the clinics, and the work that Jerome showed you and what I will try to show you is that there is an interaction, at least what we do, translational research between the clinical question coming from the bedside, and trying to answer on the bench side. So questions for the clinics are, why is the neonatal brain so prone to have seizures? Do seizures per se cause damage in the immature brain, and what are the mechanisms of the seizure-induced damage in the immature brain? Why is this important? Why we want to know the mechanism? Because so far we call antiepileptic drugs, but actually none of the antiepileptic drugs is really anti-epilepsy. They are anti-seizure, but they do not prevent the development of human epilepsy. So if we can understand the molecular and genetic basic mechanism of epilepsy, we might identify treatment that prevents not only seizures, but prevents the development of epilepsy in a determined individual. And translating discoveries at the molecular level to antiepileptic therapy, to do that, we definitely need a validated animal model. There is another issue that is coming up. So we know that most neonatal seizures tend to remain focal. So the so-called generalized tonic-clonic seizure do not really exist in the newborn. And this is because the generalization of electrical activity is actually impeded by the incomplete formation of dendrite and synapses. And so it has been shown that there is a physiological immaturity of the central nervous system because of the incomplete myelination. Fractional anisotropy, and I know really a little bit about it, is a measure that is used now in diffusion imaging that reflects the fiber density, so reflects the myelin development in the immature brain. And this is a very nice work that has been done, is still going on at UCSF by the group of Jim Barkovich, that shows with FA, fractional anisotropy, that there is a low integration of module in the newborn brain. This is a tractography in a 6-day-old boy, and actually the integration of the module is lower compared with the connectome, the adult connectome, that shows that the structural connectivity has a low integration in the newborn brain. So our question is, can seizure influence this developing network? So why do we need to work with animals? Because determining the consequences of seizure in human, as I show you, is difficult, because many factors can contribute to the outcome, and particularly the etiology, hypoxic ischemic encephalopathy, hypoxia ischemia, brain malformation, stroke. Age at onset, we have seen that there is a difference in age at onset and outcome, seizure type, frequency, duration, the use of antiepileptic drugs, and in animal studies, all these variables can be controlled. Most of the studies on seizures in the adults and in developing brain are focusing on hippocampus, because hippocampus is gray matter. Because in animals, but also in human, is most vulnerable to seizure-induced damage, because it has a very well-known role in memory and learning and memory, and because structural changes in the hippocampus, both in humans and in animals, has been shown to be accompanied by deficit in learning and memory. So why is the neonatal brain so prone to seizures? At all age, we have seizures because we have an imbalance between inhibition and excitation. So excitation is given mostly by glutamate, and again, Jerome showed you how glutamate increases in hypoxia ischemia, and inhibition is mainly given in the adult brain by GABA. But in the newborn brain, GABA is not inhibitory. GABA is actually as an excitatory function and is depolarizing. And this is a very nice work of Francis Jensen showing that in the very first weeks of life, GABA has a depolarizing activity compared with later on in the development when we'll gain the function that we'll have later on in older animal and adult animal, which is inhibition. And there is actually a sequential development of ionotropic receptor, and you can see the difference between the newborn brain and the adult brain. In the newborn brain, the GABA is mainly depolarizing, and the GABA A is depolarizing, and GABA B is silent, whether in the adult brain, GABA A is hyperpolarizing, so inhibits, so is an inhibitory neurotransmitter. So why the GABA is depolarizing? We have already glutamate. GABA is depolarizing in the human brain, in the immature brain, because one of the key factors for neuroplasticity, for development, is that neurons will connect one to each other because they get excited. And this has been shown in the past. You probably know the study that have been done in cat pups mainly. If you make them blind in the very first days of life, and then later on you make them their eyes free, their neurons in the occipital cortex won't develop. In order to develop, they need to be stimulated. So excitation is very much required in the immature brain in order to achieve a good development. So the pathophysiology of seizure, again, is that you have an imbalance between excitatory and inhibitory transmitter, but the fact is that in the immature brain you have less inhibition than in the adult brain because GABA is not inhibitory but is excitatory. So we know that the neonatal brain is prone to seizure because there is a very high level of excitation, and this excitation is required to brain development. But so do seizures per se cause damage in the immature brain? What has been shown in the adult is that in the adult animal, if you induce status epilepticus or prolonged seizure, you will see neuronal loss. So neurons, because of this high level of excitation, will die, will be replaced by gliosis. You will have a synaptic reorganization in the brain with changes in receptor function. Neoneurogenesis will take place, and this will result in behavioral deficit and the development of spontaneous seizures, which actually is a chronic disorder, which is epilepsy. Animal model of medial temporal lobe epilepsy are the most used one, and this because they show a similar pathology to the human hippocampal sclerosis of temporal lobe epilepsies, with neuronal degeneration, gliosis, axon, and synaptic reorganization, and what I will show you is the mossy fiber sprouting. Animal models of temporal lobe epilepsy have chronic hyperexcitability after an initial acute insult, after a latent period, and the animal will present with spontaneous seizure. So this is pretty much what happened in the human, in which you have a status epilepticus, then patients are seizure-free for a brief period of time, and probably during this time there is the reorganization of the synapses, and then epilepsy will start. Animal models of status epilepticus are mainly two groups: partial status epilepticus using cainic acid and pilocarpine and perforant path stimulation and kindling, and generalized animal models using fluorothil and pentylenetrozole. So what happens if you induce status epilepticus in an adult animal? This is the control. This is the CA3 region of the hippocampus, and you see that after status epilepticus, you have a marked decrease of neuronal cell in the CA2 and CA1 region that you can see very well in the animal subjected to status epilepticus. But if you do the same thing in the immature animal, this doesn't actually happen. So this is the newborn animal, this is the adult, this is the CA1 region and the CA2 region. And you see here in the adult animal after status epilepticus a huge cell loss, a huge neuronal loss that doesn't happen when you induce status epilepticus in a newborn animal. So we come up with this problem. So we know that seizures are most common in the first decade of life because the immature brain seems to be particularly prone and susceptible to seizure. But if you look at the young animals seem actually less vulnerable to seizure-induced damage. Is this true? Epileptogenesis, as well as the brain damage after hypoxia ischemia that Jerome showed you before, takes time. And this is something we are learning in the past years. It's not just the initial insult and the damage taken place here. It actually may take days to months and even to years to epileptogenesis, structural and damage at the receptor level take place. So you go from the initial insult that can be status epilepticus, can be seizures, can be hypoxia ischemia, and then you have your damage going on until you go to a chronic disorder that in our case is epilepsy. And we come up to the concept. So the main reason why we are showing you basic research here is to convey concept and convey tool to understand. So the concept of sprouting. Sprouting is an altered axonal reorganization after seizures. So what happened after seizure in adults? Those are two granule cells. This is the inner molecular layer of the hippocampus, and granule cells are actually giving excitatory inputs. In order to work well, they interconnect with a mossy cell. Mossy cell is actually inhibitory. So it modulates, the mossy cell will modulate the inputs of two granule cells that are excitatory, but they will get modulated their input through the inhibitory mossy cell. After seizures and after status epilepticus, the mossy cell die, and so the axon of the granule cell will take an abnormal connection one to each other, and because these axons were originally directed to the mossy cell, they are called mossy fiber, and they sprout. They sprout one to each other, creating a loop of excitatory cell. And you can see it here. This is an hippocampus of a naive animal, no seizure, and this is an hippocampus of an immature animal with seizure. Even in the absence of cell loss, you can see how this axon will go through the inner molecular layer and take this abnormal connection. And this is a staining that is called TIM staining, and it can label the very high content of zinc in these abnormal axons. What happened with this animal that I will show you, this alteration in the hippocampus will lead to increased seizure susceptibilities in adult animal and permanent impairment in learning and memory. So why this happened? This aberrant connection will probably permit to the epileptiform discharges in a given hippocampal lamella to recruit. It's like an excitatory loop. They will recruit additional lamellae, they will amplify the epileptic response, and they will give a persistent hyperexcitability that actually characterizes epilepsy. And so we go from an acute event, reorganization of synapses, to a chronic disease. So this is something that has been done by the group of Gregory Holmes in Gregory Holmes' lab, but for many others group later on, showing the structural and functional changes in animals subjected to status epilepticus. As many others, we used the focal status epilepticus model with lithium and pilocarpine. So these animals were given status epilepticus at a very young age, from a very early newborn period and very early infancy. And here you have an animal that has no seizures and no status, and after seizures you can see what I showed you before, so the developing of sproutings. This takes weeks or days, which can be translated into years in the human brain. This is not immediate. This takes days and weeks to develop. And this is the image I showed you before with the double staining. Those are the cell body, and those are the axon that takes abnormal connection going through the inner molecular layer of the hippocampus, and you can see it very well here as well. So after the acute insult, the brain starts to pathologically reorganize and go from a seizure to epilepsy. And then what do they do in these studies? So we looked at seizures, but we also looked at the behavioral, at neurodevelopmental. So after we induce status epilepticus, after we give seizures, then we test the animals when they are young adult, as we would do for children when they go to school. This is a testing that is very commonly used. It's called water maze, and tests the visual, spatial learning, and memory. Animals, especially rats, they have a big hippocampus, and this is very important for them to get oriented and to remember places. So animals are put in a pool, and the water is opaque by powdered milk, and this is a platform. The rats doesn't really like to be in the water, so it tries to find the platform, to get into the platform, and to remember when the platform is. So we test the animal for 4 days, and then we take out the platform, and the last day is called probe. So if the animal is smart and he learns and he remembers, he will go around, even if the platform is not here, he will go around this quadrant of the pool because he remembers that the platform was there. And so when we looked at these learning and memory testing, the water maze in those animals, those are the rats that were subjected to status epilepticus, and we have shown that they learn slowly compared with the control animal. And more importantly, when we do the probe the last day, you can see that there is definitely a significant difference between animals subjected with lithium pilocarpine status epilepticus in terms of remembering, not only in terms of learning, but also in terms of memory. And if we measure the thickness of the mossy fiber sprouting, the quantity of abnormal synaptic reorganization, there is a correlation between the thickness of mossy fiber sprouting and the cognitive impairment. So we wanted to check if this animal has also a lower seizure threshold, and we decided to use the fluorothil seizure threshold, which is a generalized, as I showed you before, a generalized model of seizures in animal. So you put the animal in a cage and you put some drop of fluorothil. Fluorothil is a potent proconvulsant that was used in the past actually at the place of electroshock, even in humans. And he induced generalized seizure in humans and animals within minutes of exposure. And what we found is actually that there was no difference between animals subjected to status epilepticus and naive animals. And this was really a problem because we thought that animals will have a low seizure threshold. So we didn't stop, and we checked in the same animal the seizure threshold using a model of partial seizure with secondary generalization. When we induce seizures with lithium pilocarpine, we elicitate seizures mainly in the limbic system. And Kindlich model is actually a stimulation of the same system that we stimulated when the animal was a newborn. So the newborn animal got seizures originating from the limbic system, and then when the animal is a young adult, we will stimulate it again, the same system. And in this case, we actually found a very higher susceptibility to seizures when you stimulate the same circuit. So the immature brain remembers the circuit that has been stimulated. So there is an epileptogenesis after status epilepticus, and this reflects age and model plasticity. Because there was a lack of effect on fluorophyll-induced seizure and a major effect on kindling, we hypothesize that there is probably a facilitation of seizure susceptibilities, that it depends on the region that has been stimulated the first time, and so gets susceptible later on. And is not actually a generalized increase of neuronal excitability, but the seizure promotes changes within a specific system. So can we modify this long-term outcome after neonatal seizures? We said about seizures takes time for the reorganization to take place, and so if we want to neuroprotect, we want to see what is this timing of places. And probably, as Jerome showed you, we are learning more and more that our time window to prevent that is probably longer than we expected in the past. In the past 20 years, 15 years, two major new concepts and new findings really have changed our way to look at changes in the immature brain after seizure. One is brain plasticity, and the second one is neoneurogenesis. When I started medical school, this was a long time ago, we thought that the brain was there and didn't change much. We could learn, but actually the structure of the brain will remain the same. And even worse, we learned that neuron after birth will start to die, and we lose neuron day by day until we get old and with little neuron left, which might be true. But anyway, so those are two very important concepts that were really new, and this is one of the first studies. This is in 2000. This is a very neat study that has been done in adults. So London is a very, very difficult place for streets, and you really need to remember and get oriented very well. 16 London taxi drivers, quite young adults, very young adults, were taken, and the average time spent on what they call the London map is also called the knowledge, was 2 years. And they studied they studied the hippocampus morphometry on structural MRI. And what did they found is that the more time they spent as a taxi driver, the bigger was the posterior hippocampus. And the posterior hippocampus is the part of the brain that is most involved in learning and memory, at the expenses of the anterior hippocampus. So there is really a structural rearrangement. The most time this taxi driver spent in learning the London map, and the most their posterior hippocampus was growing. This was very important because it shows that you can change, the hippocampus can reorganize in response to a need to store an increasingly detailed spatial representation. And that they hypothesized at that time that neurogenesis may underline these structural changes. This is done in adults, but this is very, very important for rehabilitation. This was a finding that was very, very important for rehabilitation of the developing human brain. So when we did our model in Greg Holmes' lab of status epilepticus, we decided to change cage, and we enriched this environment. This is not a normal cage. This is toys and wheels, And this is what is called enriched environment. And this is what in a human baby would be the stimulation with rehabilitation and occupational therapy. And actually, when we test with the same, this is a work of two very brilliant students from the Netherlands back in 2002 in Greg Holmes' lab, and they shown that if you put the animal in an enriched environment, this is the probe, the same water maze testing, the same type, the same model of lithium protocorpin status epilepticus, they actually do much better than animals that had status epilepticus, but they were put in normal cage, so they were not stimulated. So stimulation can change the outcome. And why this would happen? This is the second concept. This is a work that was published in 1998 in Nature by Fred Gage, and they shown that it's not true that once we are born, no more neurons, and neurons will start only to die progressively. Actually, there is a neurogenesis, a neo-neurogenesis that takes place in adult as well, and they looked at the adult human hippocampus, but this happened in other area of the brain. Neurons continue to be born. They are newly born neurons. And actually, when we looked at, when the group of, again, from Greg Holmes' lab looked at hippocampus of these environmental enriched of rats that have been put in an enriched environment, there was an increase in the newborn dentate cells in the hippocampus. This is a non-enriched environment after status epilepticus, and this is the enriched environment after status epilepticus. And this is again the work that Bridget McCabe did. And if you double label this cell with NUN, which mark for neuronal cell, and BRDU, that marks for the dividing DNA, so tells us that the cells are actually dividing, so are newly born. And you can see that those cells in the dentate gyrus of the hippocampus are actually neuron, and they continue to born and to divide after birth. So what we have learned? We have learned that prolonged seizure can cause damage in the immature brain without cell loss. They can induce synaptic reorganization and alter the neurogenesis. These can result in cognitive impairment and a decreased seizure threshold later in life. And the facilitation of seizure susceptibilities would probably reflect the seizure promote changes within a specific neuronal system. So we go back to the bench, and we go back to the crib, to the clinical side, and there are still questions. So from the bench side, neonatal seizure can cause acute effects and long-term sequelae. But I've shown you that 50% of neonates with seizure, they actually have a good long-term outcome. Recurrent seizure, they give synaptic reorganization, but we've learned that babies with benign familial neonatal seizure, which is a rare disease, but remember that rare diseases can really teach us a lot about common disease that affect many, many people. And so I show you that this is actually the first epileptic syndrome for which a gene could be localized. And the gene are KCNQ2 and KCNQ3 gene on chromosome 8 and 20. So what happened in this brain? You have this, so these two genes are mutated, and they codify, they encode for potassium channels. So potassium channels, when the cell depolarizes, potassium enters into the cell. And when the depolarization is finished, these potassium channels door open so that the potassium will go out and the cell hyperpolarize and go into rest. But if there is a loss of function, if there is a mutation in these two genes, you have a loss of function that leads to reduced potassium currents, to neuronal hyperexcitabilities, and to neonatal seizures, which is very, very important. This happens only in the neonatal period. This baby, and I will show you tomorrow, will not have seizures after the first months of life. Probably because other potassium channels will take place, and so will replace the altered function of KCNQ2 and KCNQ3. So there is an overexpression of these two potassium channels in the neonatal period. Keep this in mind, because this will be very important when we will talk about the new treatment, the new possible treatment strategies for seizures in the neonate. So what happens if you have a potassium channel active, then you have the depolarization, then the potassium channel opens, the potassium currents will start, so potassium go out and you have the repolarization. But if the potassium channel is inactive because the gene is mutated, then you have repeated depolarization, hyperexcitation, and seizures. So Nanda Singh, who is actually the geneticist that discovered KCNQ2 and KCNQ3 gene back in 1998, developed a mouse model based on the same mutation that caused benign familial neonatal seizure in human. And actually this mouse model, they very well parallel the clinical situation where recurrent seizure occur with I will show you. So Nanda Singh developed this model, animal model, and she transfected mouses with the same mutation that induced benign familial neonatal seizures. These animals were monitored by video EEG, and they shown to have multiple spontaneous, so no drugs, no lithium pilocarpine, no kainic acid, no kindling. Really, they showed to have spontaneous recurrent seizure on a nearly daily basis. Very, very similar to what happened in humans. So she looked at those rats, she really monitored those rats for seizure, and then she looked, as we did, she looked at the mossy fiber sprouting. And those are the naive animal, and those are the animal with benign familial neonatal seizure gene mutation, and they have no mossy fiber sprouting. So there is no synaptic alteration even after days and days and days of seizures. So this is really interesting because it showed us that possibly you have different mechanisms and different consequences in injured versus uninjured brain. So probably there is a difference if we give seizure, if a brain with hypoxia ischemia has seizures compared with a normal brain that has seizures because of genetic mutation. And most of the studies now are showing, and Francis Jensen lab is showing, that you can have additional damage when you induce seizure in animal with hypoxia asphyxia. There is probably a different, there are different models, even in animals, of seizure induction. There is probably a difference when you induce seizure with chemicals compared when seizures are generated by a genetic mutation. And there is a lot to learn about the time course of this abnormal plasticity that can take days, months, and maybe years. And I think this is very, very important for us, try to understand strategies and for treatment of treating these seizures. Those are all the few people that work on these works that I just showed you. Thank you. Do you have questions? Okay. Please. You should talk on the microphone because we are videotaped.
There is there are genetic markers that we can figure out that some babies or some patients will be more prone to have seizures than in certain conditions, like an acute hypoxia or sepsis, than the one who doesn't have these markers.
So I think that there are no markers for seizure susceptibility because we know that babies are susceptible to seizures because their brain is hyperexcitable. what people are looking for markers that tell you which baby will have seizure-induced damage. So the markers would be more for the development of epilepsy. And this is probably something that will happen because there is a difference. Not every baby has epilepsy after prolonged seizures in the neonatal period, and probably has to do with biomarkers, gene situation, not only in terms of which genes are bad and they make the brain prone to develop epilepsy, but probably there are some genes that are protecting the brain. And this is what we are going to do, and we are studying with rare disorders, studying the natural history of diseases in which you have a bad insult, but babies do well. So there are possibly good genes that help.
I'm here. Hi, thank you very much. I just wanted to, when I talk about those London cab drivers, I also found another paper, and it's about people who are in a plane, the stewardess. I don't know whether you know that paper. And they found that if you have a jet lag and you don't recover properly, it does have an effect on your hippocampus, and it actually is reduced in volume. And when you are in this kind of job for at least 6 years, and the thing I always want to do, but I don't get it funded, is to look at neonatologists when they start their fellowship, and then measure their volume and memory test, and then do it again when they've been in this job for like 20 years. And I think it probably will affect our hippocampus, but I think it has to be anonymous, otherwise nobody will join.
You can. I'm volunteering. Because I got jet lag from traveling from Europe and the United States. But I think it's very important. But then we should say that it might be important in the neonates in the NICU, because jet lag has to do with sleep-wake cycle. And we probably, and Nick will talk to us about that. How is it important to keep this wake-sleep cycling, that it's not always easy in the NICU. But yes, I think there are several factors, probably more than the big one we are looking for. Okay, so I think we have a break now. Thank you. Oh, another question, sorry.
Thank you very much. What is the cellular mechanism in benign familial neonatal convulsion that the epilepsy stops without treatment?
So we treat the seizures, and I will show you tomorrow. So we don't want the baby to seize too long because it's not good for him anyway. But the seizures stop after, so the seizure remits spontaneously. And this is probably because there are others potassium channels that are too immature when the baby is a newborn, and then they start to develop later on in the first month of life. So the KCNQ2 and KCNQ3 gene are still mutated, and so the potassium channel that they encode for, they won't work, but other potassium channel in the same cell will be developed, and so they will replace a little bit the ones that are not functioning. So there is a developmentally changing importance in potassium channel, but in the newborn brain, the potassium channel encoded by KCNQ2 and KCNQ3 are very, very important and relevant.
Sorry, is this mechanism of sprouting, does it happen also for electrical seizures and not, or only for clinical seizures? Because we know that normally clinical seizures are very underestimated in comparison to electrical seizures.
Supposedly, yes. There are some, most of the work that have been done in the animals, even with video EEG monitoring, they show both electrical and clinical seizures because animals they seize very much. But what, again, in Greg Holmes' lab and Nico Moshe's lab has been shown is that if you give a dose of benzodiazepine and you don't stop the seizures and the electrical discharge will continue, then you have the same damage.
Thank you. Do you have any explanation why these benign seizures do not start from birth? Why it's only at day 2 or 3 then?
Actually, this is a very interesting question. And it's an hallmark of benign familial neonatal seizure. They really don't seize right after birth. They seize after a short, free interval of a couple of 2 or 3 days. I think this probably has to be with the gene, the interaction with the potassium channel, but I think nobody looked at that. But the animals, they start to seize. The fact is that even in the work of Nanda Singh, they implanted animal after the first days of life. It's very, very difficult to implant with electrode a newborn pup after he's born. So, but basically, no, this is a very important question. I don't know why they start to seize, and I think nobody knows where they start to seize after a free period of 1 or 2 days. There is a lot to know yet. That's why it's interesting, right?
I have a question, Professor. Thank you for your talk. I was intrigued by the fact that you say that the hippocampus is the initial part of the hippocampus is the origin for a lot of these seizures, whereas I'm familiar with the fact that the hippocampus really hasn't developed that much when the baby's born at term, because in the first 18 months then the hippocampus starts developing more and more, and we know this from all the memory works, and that's why you don't really remember your own birth. And so would this, and maybe it's, I don't know if it's a question or a comment, but would this be the main influence of why the excitability of the hippocampus is so profound? Because the region has not duly developed yet? Or are there other factors there? And the other thing I'm intrigued with is that the hippocampus then seems so vulnerable, or so the plasticity of the hippocampus is so great, because we also know from research that the post-traumatic stress syndrome also affects the hippocampus quite clearly. Could you elaborate?
Thank you very much. No, this is important. So while we try to make animal models as close as possible to the human models, this cannot be perfect. And the studies I showed you, there are animal studies. And they are done in rats. Rats have a very big hippocampus. They parallel the human situation of temporal lobe epilepsy, but temporal lobe epilepsy is a situation that happens in the adults, and maybe in children with prolonged febrile status epilepticus, but not in newborns. So you are right. This is a translation of what we can see in the animals. We looked at the hippocampus because in rats it's very much developed, because it's easier, because it's very big and it's gray matter, and because of the temporal lobe epilepsy and the temporal lobe sclerosis that we have in adults. But yes, I mean, I totally agree. I mean, this doesn't parallel completely the situation of the newborn, in which probably the cortex is more important. But on the same time, I think what you're saying makes sense. So maybe it's not developed yet, but is in developing. And everything that is in developing is subjected to change if you alter the development at some point. So I think this is not developed yet, but might be developed later. But in children with prolonged febrile seizures, it has been shown that the hippocampus with MRI can have changes similar to the temporal lobe mesial temporal sclerosis in human. Okay. Thank you very much.